So, hello everyone, I am biophysics teacher from Department IT in Medicine. I am Anel Masalimova Nurjan Kızı. Today we discussed about brain potentials and the brain potential recordings. Our lesson will include three contents. The first one is introduction. In the introduction we should discuss about is biopotentials. The second is brain potentials. In brain potential, we should say about electroencephalograph because electroencephalograph is the electrical activity of the brains. The third is biopotential signals and their types. So let's start. What is the biopotentials? Biopotentials are electrical voltage generated by physiological processes in the body. Biopotentials are created by the electrochemical activity of the cell called excitable cells. When excitable cells exposed to external stimulus, it generated action potentials, which is main source of biopotentials in the body. Excitable cells types will contribute to generated of the biopotentials. The first one is a receptor or a sensory. Neurons transmit signals from the tissues and the organs to the central nervous system. The second is efferent neurons to transmit signals from the central nervous systems to efferent cells. And the third is efferent cells, which include muscle cells, and the eccentre, which stimulates physical simulations based on receiver signals. The third, brain potentials. When we started to discuss about the brain discussion, we should know for brain potentials used activity of the brain potential, we should say about electroencephalogram. What is the electroencephalogram? Electroencephalogram is a measurement of difference in electrical potential between points on the scalp. This voltage difference are results of ionic current flow within neurons and the glial cells in the brain. The electroencephalogram is usually collected in the 10 and 20 electron voltage. This arrangement of electrodes typical reference left or right hemisphere electrodes to the left and or right car lobes respectively. And the electroencephalogram can be short and long term. And we should say about that electroencephalogram can be also test that can help to diagnose epilepsy. When placing the electrodes on the head, the lead schemes used must meet certain basic requirements. The first, the scheme should represent all the main section of the convexual surface of the brain, the frontal, parietal, occipital, and the temporals. The second is maintained above one of the main characteristics of a normal electroencephalogram. It is significant symmetry. The electrodes must also be placed symmetrical about mid-sagittal lines of the head. Thirdly, it is since the potential difference depends on the distance between the electrodes, and the distance between the electrodes must be same. The second picture, if we consider that the lead of the record potential difference between two electrodes, then we should say that depends on the electrode leads divided two types. It's a monopolar and bipolar. Monopolar, what is a monopolar? In the picture we see the first picture. It is a bipolar electroencephalogram derivation. The potential difference is measurement between two separate electrodes, and that is all electrodes are divided into pairs and the signals is recorded from each pairs. The second is monopolar, and the second picture monopolar. What does this mean? Monopolar electroencephalogram derivations. The potential difference is recorded between active electrodes and the common reference electrodes. And to register electroencephalogram, we should need to separate the possibility points to the location of electrode intervals 10% and 20%. In this slide, we see between these frontals and the centrals occipitals electrodes, distance should be 20%. Between the nasions and the anions, it should be 10%. And you see in this picture. Electroencephalogram, we should say that it is a test to brain wave. And to describe the brain wave, electroencephalogram's rhythms can be divided 5 types rhythms: alpha, beta, gamma, theta, delta waves. What is alpha waves? Alpha waves is considered moderate rhythms and usually seen in the occipital regions on the head. Beta waves is considered fast rhythms, and the beta waves typically observed on both sides of the head and the most even the frontal. Beta waves can be substantially reduced in areas of cortical damages. And the third is delta waves. Delta waves is considered slow rhythms and tend to be the high amplitudes. Delta waves are normally dominate rhythms in infants. Theta waves, third waves, theta waves, it is considered slow rhythms, 4 and 8 Hz. Theta waves are normal in children up to 13 years old. Theta waves are found in sleeping adults, and the consistent theta is considered abnormal in conscious adults. Gamma waves, the last rhythms, the gamma waves. And gamma waves is considered the fast rhythms, and the gamma waves are present during the mixed sensory processing, such as perceptual tasks as a combined hearing and seeing. And so next, in conclusion, we can say that electroencephalogram can be useful to determine coma and brain days and evaluate inhibitor control over behavior and establish brain-computer interface and monitor sleep physiologies. And electroencephalography can be also evaluated anesthesia depth, locate seizure origins, and test drugs effects for convulsions and seizures. And also can be used to assist in cortical existence, to surgery, for example, of epileptic lockups. Thank you for your attention.